19%. That's the number. Just sit with it for a second before I tell you what it means, because once I do, the number changes shape entirely. Los Angeles, February 12, 2020. A geneticist named Sriram Sankararaman is sitting at a workstation in UCLA's Department of Human Genetics, looking at a genome-wide ancestry map of 405 West Africans. He has been running this analysis for months. The lab smells like recycled air and old coffee. There is, by all accounts, nothing unusual about the scene, except for what is on his screen. Up to 19% of the DNA in these genomes, Yoruba people from Ibadan, Nigeria, comes from a species that has never been found. No fossil, no skull, no name. No site where anyone has ever dug up a bone and said, here it is. The species exists only as a number on a screen. 19%. Sankararaman and his colleague Arun Durvasula had compared those 405 West African genomes against the only 2 archaic human reference genomes science actually possesses, a 44,000-year-old Neandertal from Eastern Europe and a Denisovan from Siberia. The archaic segments in the Yoruba DNA matched neither. They were older, they were something else entirely, and there was no physical evidence anywhere on Earth to tell them what. Stay with me because this next part is the reason this video exists. You probably know about Neandertal DNA in European genomes. That story has a fossil. It has a cave in Croatia. It has bones that a team extracted ancient DNA from and sequenced. The Yoruba's archaic relatives have none of that. No cave, no bones. The evidence exists only inside living cells. And it has been there, undetected, for the entire time that scientists were using Yoruba DNA as their definition of what a normal, uncontaminated modern human genome looks like. That's the part that stopped me cold. For 2 decades, the Yoruba from Ibadan have been the most important baseline population in global genomics. Every disease study, every drug trial, every calculation of how much archaic DNA other populations carry. When researchers needed a 0 point, a clean, uncomplicated modern human genome to measure everything else against, they used the Yoruba, and the baseline was never 0. It was the most complicated genome in the room. Here's what that number on Sankararaman's screen actually means. Somewhere between 2 and 19% of the Yoruba genome was inherited from a species that diverged from the human line over 1 million years ago. A species that left no bones in any museum, no tools in any archive, no name in any record. Only this number, only this signal. Only these 53 million living people who carry it right now and have no idea. If you've ever actually wondered where human beings come from, not the polished version, but the real biological answer, you're going to want to subscribe. Because what comes next goes deeper than most people are willing to go. The species whose DNA lives in these genomes diverged from the human line over 1 million years ago. And no one has ever found a single bone. So, if the species has no bones, how do we know it was real? That's the first question. And it's a good one. Because in science, you don't just get to say something was there without evidence. Sankararaman and Durvasula knew that. They built a method specifically designed to find archaic ancestry without any reference genome to compare it to. And what that method revealed was this. The archaic segments in Yoruba DNA are on average 1.69 times older than the non-archaic segments in the same genome. 1.69 times older in the same body. That age gap cannot be produced by 2 modern human populations diverging and then reuniting. The mathematics require something categorically older, not a distant cousin. Something that had been separate for a very, very long time. The divergence estimate is staggering. This ghost population's ancestors split from the lineage that would eventually produce both modern humans and Neanderthals somewhere between 360,000 and 1 million years ago. To put that in human terms, when this split happened, Homo heidelbergensis was the dominant hominid in Africa. The creature whose DNA is now inside living Yoruba people was already a separate species before our most recent common ancestor with Neanderthals had even emerged. Before, we were not yet what we are. They already were something else. But here's where it gets strange. We have no bones, no tools, no sight, nothing physical. And that isn't because the species didn't exist. It's because of where they lived. The West African forest, dense, humid, hot, and ancient beyond description. Tropical forest doesn't preserve organic material. A bone left on that forest floor in 200,000 BCE doesn't survive, it dissolves. The humidity, the bacteria, the chemistry of the soil, all of it conspires to return biology to the earth inside a few thousand years. The fossil record in West Africa isn't silent because nothing happened there. It's silent because the forest erased everything that happened. Every absence in the African ancient DNA record is not a chapter that didn't happen. It's a chapter written on material that didn't survive long enough to be read. By 2020, no ancient West African hominin fossil older than about 16,000 years had been successfully sequenced. The physical record had been reclaimed. What remained was the sequence, nested inside 53 million living people, waiting for a method sensitive enough to find it. Here's the part that makes this bigger than one population story. The ghost introgression window. The period during which this unnamed species was interbreeding with the ancestors of the Yoruba runs from approximately 124,000 years ago all the way to zero. That window includes the out-of-Africa migration, which means some of this signal didn't stay in Africa. Some of it left. Actually, hold on a second. Before we go further, I want to ask you something directly. If you've taken a consumer DNA test, your results probably show your African ancestry as a percentage with a regional label. After what I just told you, do you think that label is telling the whole story? Drop it in the comments. Your actual percentage and what you think it's actually measuring. Because honestly, after the next few segments, your answer is going to hit differently. January 22, 2008. The National Institutes of Health in Bethesda, Maryland, puts out a press release. It announces the 1,000 Genomes Project, the most ambitious map of human genetic variation ever attempted. 26 populations, over 1,000 individuals. The most detailed picture of human genetic diversity ever assembled. One population appears first in every announcement. Yoruba in Ibadan, Nigeria. They would become the reference, the anchor, the definition in practice of what an uncontaminated modern human genome looks like. And for 12 years, that is exactly what they were. And yet, the data said the exact opposite of what everyone assumed. Here's how that happened. When researchers want to measure how much Neandertal DNA a European person carries, they need a comparison point, a population with zero Neandertal admixture, so they can calculate the difference. The Yoruba were chosen for exactly that reason. Every comparison ran through them. Every calculation of archaic ancestry in non-African genomes used Yoruba DNA as the zero. If the Yoruba baseline carried its own archaic signal, every single calculation built on top of it needed revisiting. The 1,000 Genomes Project, completed in 2015, identified over 88 million genetic variants across those 2,504 individuals. The Yoruba cohort from Ibadan, catalogued as YRI in the database, became the most cited African reference in medical genetics globally. Thousands of genome-wide association studies, cancer research, diabetes, cardiovascular disease, all of it anchored to data from Ibadan, all of it assuming the baseline was clean. The baseline wasn't wrong, it was under-resolved. That's the distinction that matters. The researchers who chose the Yoruba weren't making an error in judgment, they were using the best tools available. The ghost signal in the Yoruba genome was invisible to the methods of 2008. It was invisible to the methods of 2015. Durvasula and Sankararaman's 2020 method was the first one sensitive enough to detect it. The Yoruba weren't misrepresented. The instruments weren't good enough yet to see what was there. But here's the consequence that nobody talks about. The 216 Yoruba individuals whose genomes drove the 2020 discovery had given blood samples in Ibadan years earlier because researchers told them it would help map human disease. They were right. It did. It just also mapped something that predated human disease by 1 million years. Those same samples, sitting in the National Center for Biotechnology Information Database, freely accessible to any researcher in the world, those samples contained a species science had never found, and nobody knew until the right question was finally asked. The Yoruba were chosen as the world's genomics baseline because their DNA appeared to be the most modern. The 2020 finding inverts that precisely. They are the population carrying the oldest non-modern signal on the planet, not instead of being the baseline. While being the baseline. Both things are true. And that is where this story gets genuinely hard to hold. East African Rift Valley. Approximately 41 to 400 years ago, a mutation occurs on the Y chromosome of a single man. He has no idea. Why would he? The mutation is silent. It doesn't change his face, his strength, his lifespan. But it is specific enough, stable enough, heritable enough that 400 years later, it sits in the Y chromosome of 93.1% of Yoruba men from Ibadan to Lagos to London to Atlanta. A haplogroup called E1B1A, the marker that traces every Yoruba paternal line back to one moment in East Africa, before agriculture, before writing, before the concept of a bloodline as something worth recording. This was a discovery made by researchers mapping the Y chromosome landscape of Niger-Congo language speakers. A study that confirmed the haplogroup's highest diversification point, its most ancient branching in living populations, sits in the Yoruba from southwestern Nigeria. The estimated time to most recent common ancestor for this specific subclade, E1B1A7A, in the Yoruba, approximately 4,200 years. That's when the paternal lines that lead to every living Yoruba man converge to a common point in what is now Nigeria. That mutation 400 years ago is alive in the world right now. It's in the Y chromosome of a man working a double shift somewhere in Lagos, in a doctoral student in London who's never thought about this, in a truck driver in Atlanta whose father's father's father, 1,656 generations removed, carried the same single letter in his genome, same letter, 41,000 years. But here's where the story takes a turn that the shipping records never accounted for. Researchers at 23andMe, led by Stephen Micheletti and a team that spent years pairing tens of thousands of genetic profiles with detailed historical records, found something that directly contradicts what the transatlantic shipping manifests say. Among US-based African Americans, the largest single source of African ancestry is not what those manifests would predict. It's Nigeria, specifically the Yoruba, the Isan, and the Igbo. The dominant genetic signal in the African American genome points to the Bight of Benin and the Niger Delta, not to Senegambia, not to Sierra Leone, despite historical records suggesting those regions were more heavily trafficked to North America. The genome disagreed with the manifests, and the reason is secondary slave trading. Enslaved Yoruba people were transported first to the Caribbean, then moved north within the Americas. The manifest tracked the first voyage. The genome recorded the full story. The official written history of where African Americans come from was maintained by people who had a financial interest in incomplete record keeping. The biological record had no such interest. It simply preserved what happened. This pattern keeps appearing. And I mean this sincerely, not as a transition device. We've now seen it twice. A population whose written history was controlled by others, while their genome kept its own record. If you've been watching this channel for a while, you've seen this same conflict between archive and biology in other populations too. Which do you trust more, the written record or the biological one? Tell me in the comments. And why? Because the answer says something about how we think history belongs to people. The Bight of Benin, that stretch of coastline directly offshore from Yoruba territory. Between 1650 and 1807, more enslaved Africans departed from this region than from almost any other single point on the continent. The slave traders called it the Slave Coast. They kept records of tonnage, of voyage durations, of port fees. The things they did not record, could not record, were the haplogroups, the mitochondrial sequences, the cellular inheritance that would survive long after every manifest had yellowed and crumbled. The 23andMe study I mentioned, Micheletti and colleagues, published in the American Journal of Human Genetics in 2020, found something in the genomic legacy of the transatlantic slave trade that stops most people mid-sentence when they hear it. In parts of Central America, enslaved African women contributed 13 to 17 times more to the present-day gene pool than enslaved African men. 13 to 17 times. For North America and the Caribbean, the female contribution was consistently, significantly higher than male across the board. This isn't just a number. This is a measurement of who died. Enslaved African men died at higher rates. They were worked to death in higher numbers. They were killed in higher numbers. Their Y chromosome lineages were more thoroughly erased from the living record. What survived, disproportionately, was the mitochondrial DNA, the maternal line. The cellular inheritance, passed from mother to child, carried in the small circular genome inside every mitochondrion in every human cell. And mitochondrial DNA is inherited exclusively through the maternal line. This isn't just ancient history. This is in your DNA right now, if you have African or African American ancestry. The female line is where the record survived. Now here's what makes this specific to the story we've been building. Because Durvasula and Sankararaman's 2020 analysis mapped the ghost archaic signal in the Yoruba genome, and because the archaic segments they identified cluster in, among other places, the mitochondrial lineage that runs through the maternal line, the sex bias in middle passage survival rates becomes something more than history. It becomes a genomic preservation mechanism. The most ancient non-modern DNA signal in any living human population survived 3 centuries of the most violent forced migration in recorded history. It survived because the women who carried it in their cells were slightly more likely than the men to reach the present genetic record intact. Not because anyone planned it, not because the biology knew what was at stake, but because the mathematics of survival and inheritance aligned in one specific way and the signal made it through. That window, during which the ghost species was interbreeding with Yoruba ancestors, 24,000 years to 0, includes the entire span of recorded human history. The ghost was still contributing to the human gene pool, while other civilizations were building their first permanent structures. And it almost didn't make it to the present. Almost. Before we go any further, I want to give the strongest possible argument against everything I've told you in the last 20 minutes. Because it exists, it's rigorous, and you deserve to hear it. The argument goes like this. West Africa has been inhabited by anatomically modern humans for at least 300,000 years. During that time, multiple population lineages diverged, lived in isolation from each other for tens of thousands of years, and then eventually reconnected and mixed. No archaic hominins required. Just ancient, modern humans, separated for long enough that their genomes look old and diverged when you run today's analysis tools across them. The signal Durvasula and Sankararaman found could be ancient population structure, not genuine introgression from a pre-Neandertal species, but the remnant of a deeply diverged modern human population that simply looks archaic in comparison. This is not a fringe objection. Pontus Skoglund, one of the architects of African ancient DNA methodology at the Francis Crick Institute in London, raised exactly this as the interpretation that needed to be ruled out. It's careful, it's precise. It's the kind of question that separates serious science from sensationalism. What they found next changed everything they thought they knew about this. Durvasula and Sankararaman ran the test. They specifically modeled the population structure hypothesis, asked whether ancient divergence within modern humans could produce the patterns they were seeing. The answer came back from the data, not from their preference. The archaic segments in Yoruba DNA are on average 1.69 times older than the non-archaic segments in the same genome. Ancient population structure, two groups of modern humans diverging and later mixing, cannot produce that age gap. The mathematics of population genetics are specific here. To generate a 1.69 times age differential within a single genome, you need a lineage that was genuinely separated for an extraordinary length of time, not a distant cousin, something categorically older. For context, the age differential between Neandertal introgressed segments in European genomes and non-introgressed segments is in the same range, the same test, the same signature, applied to Yoruba genomes, producing a comparable result. The skeptics model doesn't balance the equation. Here's what Sankararaman said in the week the paper published. He said, "We don't need reference DNA from fossils of the archaic hominid to confirm that, somewhere deep in our ancestry, humans intermixed with them. That's not confidence for its own sake. That's a man describing what the math supports without a single physical bone in hand. The debate isn't closed. Papers are still being written. Not every researcher has signed onto every implication. But the population structure hypothesis has been tested against the data, and the data has pushed back. What remains is a ghost that the statistics confirm and the fossil record cannot provide. Because the tropical forest, as we said, doesn't preserve what it swallowed. What is no longer in question is the scale of what the Yoruba genome contains. And the debate over its precise origin is, in a way, the least surprising thing about it. Because the ghost is in there, the argument is only about what to call it. Right now, in a cell dividing somewhere in Ibadan, a sequence is being copied. It has been copied for 124,000 years. The Muslims don't know its history. The proteins don't care where it came from. They just read it and do their work. Among the sequences being read is one that came from a species that has no name, and it is not dormant. It is not vestigial. It is not some fossil remnant sitting inert in the genome like a dead star in a night sky. It is working. Everything I just told you, hold on to it, because it's about to mean something completely different. Durvasula and Sankararaman didn't just find the ghost signal. They ran it through natural selection filters, methods that identify which genetic segments have been actively retained because they conferred survival advantages. The archaic segments that pass those filters in Yoruba genomes cluster around genes associated with tumor suppression and hormone regulation. The ghost wasn't merely tolerated by the immune system of the ancestors who carried it. It was kept, selected for, passed forward across 124,000 years of human generations because it helped the people who had it to survive. I want to be precise about what that means and what it doesn't mean. The tumor suppression connection is statistically significant in the published data. The specific pathways overlap with cancer resistance mechanisms already known to science. The research is ongoing. This isn't the final word, but the signal is there, and it's pointing at something that human biology found useful enough to preserve for over 100,000 years. That is not a small thing to say about the DNA of a species that left no other trace. We have been calling it archaic DNA throughout this video. Here is a more accurate description. It is functional inheritance from a population that survived something we don't understand, passed that survival forward to our ancestors, and those ancestors carried it through every catastrophe and migration and bottleneck that followed, all the way to 2020, when two scientists in Los Angeles finally had a method sensitive enough to see it. Now, here is the part that I've been building towards since the first segment. The ghost signal survived because Yoruba women, specifically, were the primary carriers of the archaic mitochondrial sequence, and the transatlantic slave trades documented sex bias, enslaved African men dying at higher rates, contributing disproportionately less to the surviving gene pool, meant the female line was more likely to reach the present genetic record intact. The most ancient non-modern DNA in any living human population was nearly erased by a modern human atrocity. And it survived because women who were enslaved, stripped of every other record, transported across an ocean against their will, carrying in their cells a sequence from a species that predated the human concept of recorded history, those women made it through. Not because anyone protected them, not because the biology knew what was at stake, because they were slightly more likely to survive than the men beside them. That is the margin. That is the thread. There is a woman alive today in Lagos, in Brooklyn, in London, who carries in her mitochondrial DNA a sequence inherited from a species that diverged from the human line over 1 million years ago. She does not know this. It is functioning. It has always been functioning. In 2020, two things happened on opposite sides of the Atlantic at roughly the same time. Researchers in Nigeria were conducting genomic sampling of living populations, and researchers at 23andMe were matching the DNA of hundreds of thousands of African Americans to West African source populations. 2 studies, 2 continents, 1 story. And in both of them, the Yoruba kept showing up as the dominant genetic thread, more present in the diaspora than the shipping records predicted, more intact across 4 centuries of disruption than almost any demographic model would have suggested. There are 3 things the combined data revealed. The second one surprised the researchers. The third one changes the way you think about what a genome actually is. First, Nigerian ancestry, Yoruba, Isan, and Igbo together, is the largest single contributor to present-day African-American genomes, not Senegambian ancestry, not Sierra Leonean ancestry. Despite historical records indicating those populations were more heavily trafficked directly to North America, the genome corrected the archive. Second, the reason is secondary slave trading. Yoruba-speaking Africans were transported first to the Caribbean, then moved north within the Americas through an internal trade that the transatlantic shipping records largely don't capture. The manifest tracked the first crossing. The genome recorded every step after. Third, and this is the 1,000 Genomes Project's Yoruba cohort from Ibadan remains, today, the primary African population reference for biomedical research worldwide. Every drug trial, every genome-wide disease study, every pharmacogenomic analysis that uses an African anchor is using data from Ibadan. The genome that carries the oldest non-modern signal on the planet is also the one underpinning the medical research that will determine how drugs are designed for African and African-descended populations for the next generation. The most complex genomic story in human prehistory is sitting at the foundation of 21st century precision medicine, and most of the researchers using it still don't fully know what's in it. Micheletti and his team said it plainly in their 2020 paper. The genomic legacy of the transatlantic slave trade tells a story that the written record cannot. The shipping manifest lists objects. The genome names people. And not just people, it names lineages, survival patterns, the specific shape of who made it through and who didn't, measured in haplogroup frequencies that no one could falsify and no one could burn. That's what the genome is. Not just ancestry, not just medicine. It is the record that survives when every other record has been taken away. What would fully resolve the ghost archaic question? The one piece of evidence that would allow science to name the species and map its biology is an ancient West African hominin fossil with enough organic preservation to yield a reference genome. Without it, the ghost remains definitionally unnamed. The tropical forest that swallowed all previous fossils shows no sign of releasing this one. The absence is structural. The question stays open. A single cell dividing. This is happening right now, in Ibadan, in Lagos, in Brooklyn and London and Sao Paulo. The sequence being copied has been copied continuously for 124,000 years. It came from something that no scientist has ever held, no museum has ever cataloged, no textbook has ever named, and it is still here. If your ancestry includes West African or African American lineage, and for a significant portion of people watching this, it does, there is a measurable probability that somewhere in your mitochondrial DNA, there is a sequence your testing company labeled West African. What they were measuring is one layer, one resolution. Beneath it, in the places the instruments of 2008 and 2015 couldn't see, there may be something older, something that your testing company has no category for, because science has not yet found the fossil that would let it create one. You are not just descended from Africa. You may be descended from something that preceded Africa's own deep recorded history. And the only reason we know this exists, the only reason this signal made it to the present, is because of the women who carried it through the worst thing that ever happened to them. The ghost archaic species has no name, no location, no bones in any collection anywhere on Earth. It has one thing: 53 million living witnesses. The Yoruba, and everyone who carries Yoruba ancestry anywhere in the world, are the evidence for a species that left nothing else behind. Science found it by looking in the last place it would have looked. The most studied genome on the planet. The one that was supposed to be the baseline. The one that was supposed to be zero. If you want to understand what else is hidden in the genome science thought it already understood, subscribe. Because the Yoruba genome is not an exception. It is a warning about every other baseline we've trusted, without looking underneath. 53 million people carry the only biological record of a species that lived and died before the concept of a name existed. The shipping records didn't list them. The fossil record can't find them. The genome still has them. It has always had them.